Hello and welcome back to the series of videos of the metabolism. Today we're going to focus on the nitrogen metabolism. In particular, in this video we're going to discuss the degradation of amino acids. So let's begin. Okay, so we are here inside a cell. This is a muscle cell, for example, and we have an amino acid. In this example we are going to use alanine, and I will explain why later. Alanine and alpha ketoglutarate. Do you remember the metabolism of the Krebs cycle? This is a metabolite in the Krebs cycle. So these two, this is an amino acid because it has an amine group, and this is the alpha keto acid because it has a keto group in the alpha carbon, the carbon number 2. They are going to bind together and they are going to swap the groups. The alanine is going to give the amine group to the alpha ketoglutarate, and the alpha ketoglutarate is going to give the keto group to the alanine, and we get pyruvate and glutamate. Okay, this reaction is called transamination. It's very important because once we digest those proteins in the diet and we get them in our cells, we get those amino acids and we want to degrade them because we need energy, and transamination processes are very important for that to happen. We want the amine group from the amino acids to go to the glutamate. Glutamate has a very important role. He's going to get all the amine groups from pretty much every amino acid and is going to store it. Glutamate is very, very important. So to catalyze this process, to make it faster, we have, of course, an enzyme. We have the GPT enzyme, which stands for glutamate pyruvate transaminase, and the same enzyme can be called alanine amino transaminase. So we need the pyridoxal phosphate. Pyridoxal phosphate is the active form of vitamin B6. If you don't know what this is, check my video on vitamins in the nutrient description series. Okay, so alanine is basically going to give this amine group to the pyridoxal phosphate. At the same time, the alpha ketoglutarate is giving to the pyridoxal phosphate the keto group, and then the pyridoxal phosphate is going to deliver those amine and keto group to the molecules, as you can see from the arrows. This is the transamination. Okay, so once we have the pyruvate, in this case, you know, pyruvate can go and create acetyl-CoA, as we talked in other videos, but pyruvate can also go and form lactate, and you know that lactate is going to go to the blood and then to the liver. This is the liver, and that's called the Cori Cycle. The Cori Cycle gets the pyruvate, converts it into lactate, and then lactate travels to the liver to be converted back to pyruvate and then go for gluconeogenesis. Glucose then is exported back to the muscle to be used. This is a mechanism the muscle and the whole body uses to get new glucose from pyruvate. We have to mention that alanine can help this process of getting new glucose because alanine can be synthesized from pyruvate, and we're going to see that, and alanine can travel to the blood and then enter the liver, and that's the glucose-alanine cycle. And as the lactate, alanine can be converted back into pyruvate that can go through gluconeogenesis. Okay, so you know, we talked about glutamate and its responsibility on getting all the ammonia, all the amine groups from the other amino acids and to store them in the cell. And this is the representation of all the tissues, but in depth we're going to focus on astrocytes in the brain. Glutamate can be converted into glutamine. Now, glutamine is very important because glutamine is the form of glutamate with another nitrogen, with another ammonia, with another amine group, and glutamine can travel to the blood and get inside the liver. These two amino acids are very, very important because one is in charge of recollecting all those amine groups from the other amino acids from the transaminations, and then it passes those amine groups to the glutamine to send them to the liver. This process of creating glutamine from glutamate is done by the glutamine synthetase and requires ATP. So this is a regulation of glutamine synthetase, and it's very tricky, but it's simple once you understand it. Okay, so this is the GS, the glutamine synthetase, which is active here, and we have an adenylate transaminase, which we are going to name as ATase, and the protein 2. Now, here's the thing. We have the glutamine synthetase and an adenylotransferase. The adenylotransferase is going to put AMPs to the glutamine synthetase to inactivate it. And we have the protein 2. The protein 2 is going to stimulate this step to occur. But if the protein 2 is bound to the UMP, it will do the reverse. It will inactivate the ATase and it will remove the AMPs, activating the glutamine synthetase. So from glutamate we create glutamine. But here's the thing. In the brain, if we have too much glutamine, it's probably due to the fact that we have too much ammonium. Ammonium is toxic. And ammonium is going to be converted into glycine. And these two metabolites, they act as electrolytes in the astrocytes in the brain. And as being electrolytes, water will follow their concentration, and this will cause edemas because the water is going to get inside. Okay, so we talked about how glutamine will get those amine groups and send them to the liver. But why we didn't mention that in the muscle? Well, because as being a synthetase, we need ATP to create glutamine from glutamate. And we don't want that in the muscle because the muscle needs that energy to contract. So they have another mechanism. The muscles use alanine instead of glutamine. And alanine, as I said, can go to the liver. And it will be used for both things, glucose-alanine cycle and the recollection of amine groups as well. Now, I mentioned this enzyme here, the alanine aminotransaminase. This is one of the most important ones. The other important transaminase, we're going to see it now. Because if we follow glutamate, glutamate can travel to the blood and then inside the liver. We're inside the liver here. And then it's going to enter the mitochondrion. And inside the mitochondrion, we have the oxidative deamination. This is not a transamination, this is a deamination. That means we are going to get rid of an amine group. This amine group is going to create ammonia. The enzyme we're going to use is the glutamate dehydrogenase. The glutamate dehydrogenase needs the NADP positive and is going to create NADPHs. These are used in the reaction against free radicals. We're going to use water as well, and then we get the formation of alpha-ketoglutarate from glutamate. We have the alpha-ketoglutarate again and aspartate. This is another amino acid. And this is the second most important transaminase, the aspartate amino transaminase, or GOT. That stands for glutamate oxaloacetate transaminase. So we need a keto acid and an amino acid. We have aspartate and alpha ketoglutarate. They're going to bind together as before. Aspartate gets bound to the enzyme and then it's going to give its amine group to the pyridoxal phosphate, and then pyridoxal phosphate is going to give it to the alpha ketoglutarate, creating glutamate. At the same time, the alpha ketoglutarate is going to give its keto group to the pyridoxal phosphate, which will deliver it to the aspartate, creating the oxaloacetate. Now, if we continue, we have amino acids degradation. Okay, if we start here, pyruvate. This is a common pathway of the Krebs cycle, the transition step. Indeed, we can put glycolysis here if we want. Basically, here we have these amino acids will give pyruvate. Their degradation will give pyruvate. From cysteine, we get the pyruvate. From the tryptophan, we get the alanine. And then the alanine is going to give us pyruvate. From the threonine, we get the glycine. And the glycine is going to be converted into serine. And the serine will give us pyruvate. Here we have the amino acids that will give us either acetoacetyl-CoA or acetyl-CoA. These are called the ketogenic amino acids. The threonine, the tryptophan, the isoleucine, and the leucine will give us acetyl-CoA directly. So if we transaminase those amino acids, their carbonated skeletal will give us acetyl-CoA. And then we have lysine, phenylalanine to tyrosine, and those two acetoacetyl-CoA, as well as the tryptophan and the leucine. This molecule right here, it's the precursor of acetoacetate and acetone and beta-hydroxybutyrate, the ketone bodies. Now, if we continue, we have the asparagine that is going to be converted into aspartate, and the aspartate, as we saw in the transaminase enzyme we used earlier, is going to give us oxaloacetate. The phenylalanine is going to be converted into tyrosine. As you can see here, is the same. And the tyrosine will give us fumarate as well. Now, methionine is going to be converted into homocysteine and then to alpha ketobutyrate in this case, and then to propionyl-CoA. This is methylmalonyl-CoA. You probably know these two if you saw the video about the fats. And then succinyl-CoA is created. As well with the isoleucine to propionyl-CoA, valine to propionyl-CoA, and threonine to alpha ketobutyrate to propionyl-CoA, methylmalonyl-CoA, and succinyl-CoA. And they will continue the Krebs cycle. And here we have glutamine to glutamate. That's what glutamine does inside the liver. Is going to be converted back into glutamate, and then glutamate to alpha ketoglutarate, as we saw in the transaminase enzymes. Proline to glutamate gamma semialdehyde, and then to glutamate. Histidine to glutamate directly, and arginine to ornithine. This is a metabolite of the urea cycle as well as arginine. We are going to see that in another video. They go to the glutamate gamma semialdehyde and then to glutamate. And this is the amino acid degradation. We have to mention that the branched chain amino acids are not degraded in the liver. So for isoleucine, leucine, and valine, this is not going to happen in the liver, but in other tissues, yes. And these are the monocarbon transfers we're going to use. Biotin is going to transfer the carbon, the monocarbon, in form of CO2. Then we have the tetrahydrofolate, which is the active form for the vitamin B9, and the monocarbon groups in the intermediate oxidation state, and some methyls. So this is going to help us with some methyls and the normal carbons we have. And then the most powerful one we have as monocarbon transfer is the S-adenosylmethionine that will transfer methyls directly. These are going to modify those structures we get from the first transamination of the amino acids. Okay, that's it for this video. I hope you liked it. Like the video if you did. Subscribe to the channel. Comment down below if you have any doubts or suggestions. I will leave this diagram in the description so you can have it. It's yours. Study it very well and see you on the next video. Bye.